structure and function of ear-air bladder connection with Weberian apparatus in fishes, different types of caudal fins, modification of caudal fin, origin of the paired fin. Air bladder, swim bladder. In fishes, the air bladder, also known as the swim bladder, is a gas-filled sac present in most bony fishes, osteichthyes. It plays a key role in maintaining buoyancy, allowing the fish to remain at a desired depth without expending energy. In addition to buoyancy control, it is involved in sound production and reception in some species. Structure of air bladder. The swim bladder is typically located in the dorsal portion of the body cavity and varies in structure depending on the species. It is composed of 1. Outer wall, a tough, elastic connective tissue. 2. Inner layer, lined by a gas gland responsible for secreting gases. 3. Rete mirabile, a capillary network that helps in gas exchange. 4. Oval, or resorptive area, a region where gases are released to reduce buoyancy. Types of air bladder. Based on the presence and functionality of the duct connecting the swim bladder to the esophagus, there are two major types of swim bladders. 1. Physostomous swim bladder. Structure. Contains a pneumatic duct that connects the swim bladder to the esophagus or gut. Gas regulation. Fish can gulp air from the surface to fill the bladder and release it through the mouth or gills. Example: goldfish, catfish, and eels. Significance: allows rapid adjustment of buoyancy but requires access to atmospheric air. 2. Physoclistus swim bladder. Structure: lacks a direct connection to the esophagus. Gas regulation. Gas exchange occurs through specialized blood vessels, rete mirabile and gas gland. Example: perch, cod, and cichlids. Significance: more advanced mechanism allowing better buoyancy control in deeper waters. Mechanism of gas secretion and absorption. The regulation of gases within the swim bladder is crucial for maintaining neutral buoyancy and adjusting to different depths. Gas secretion, inflation. 1. Gas gland. The gas gland plays a central role in secreting gases into the swim bladder. 2. Rete mirabile. A countercurrent exchange system of capillaries helps concentrate gases, primarily oxygen, in the blood near the gas gland. 3. Bore and root effects. Hemoglobin's ability to release oxygen is influenced by pH, Bohr effect, and the presence of high concentrations of carbon dioxide. Root effect. 4. Lactic acid secretion. The gas gland secretes lactic acid, lowering blood pH, reducing hemoglobin's oxygen affinity, and facilitating oxygen diffusion into the swim bladder. 5. Gas diffusion. The concentrated oxygen from the blood diffuses into the swim bladder across the gas gland membrane. Gas resorption, deflation. 1. Oval body. A specialized area where the bladder wall becomes thin, allowing gas release into the blood. 2. Sphincter muscle. Controls the opening of the oval to regulate the release of gas. 3. Venous circulation. The released gas is carried away by blood and expelled through gill respiration. Functions of the swim bladder. 1. Buoyancy regulation. Helps maintain neutral buoyancy at different depths. 2. Hearing. In some fishes, it is connected to the inner ear via the Weberian apparatus. 2. Enhance sound reception. 3. Sound production, used for communication or courtship in certain species by rapidly contracting muscles around the swim bladder, e.g., in croakers. 4. Respiration. In primitive fishes in some species, the swim bladder functions as a respiratory organ, providing oxygen exchange similar to lungs. 5. Hydrostatic organ. Pressure detection. The air bladder can help detect changes in water pressure as a fish moves vertically in the water column. 6. Body stability and posture. The position and size of the swim bladder influence a fish's center of gravity, aiding in body stability and balance. 7. Pressure equalization in the inner ear. In some species, the swim bladder helps in equalizing pressure in the inner ear, aiding in maintaining equilibrium and orientation. Summary of functions. Function: buoyancy regulation. Description: maintains vertical position in water without expending energy. Examples: perch, trout. Function: sound reception. description enhances hearing through vibration transmission. Examples: carps, catfish, function sound production, description generates sounds for communication or defense. Examples: croakers, catfish, function respiration, description acts as a lung-like organ for breathing atmospheric oxygen. Examples: lungfish, gar, function hydrostatic pressure detection, description helps detect depth-related pressure changes. Examples: most teleosts, function body stability and posture, description affects balance and stability in water. Examples: cod, bass, structure. Of Weberian ossicles in fishes, the Weberian ossicles are a series of specialized, small bones in fishes belonging to the superorder Ostariophysae. These ossicles form part of the Weberian apparatus, a unique evolutionary adaptation that connects the swim bladder to the inner ear. This system enhances the ability to detect sound vibrations in the water, improving hearing. Components of Weberian ossicles. The Weberian ossicles are derived from modifications of the first 4 or 5 vertebrae in the fish's spine. These bones form a chain that transmits vibrations from the swim bladder to the inner ear. The key components are: 1. Claustrum, the smallest and most anterior ossicle, located near the back of the skull, helps connect the swim bladder to the inner ear. 2. Scaphium, positioned immediately behind the claustrum, contributes to transmitting mechanical vibrations. It articulates with other ossicles and forms part of the linkage to the sensory structures of the ear. 3. Intercalarium, lies between the scaphium and tripus, acts as a relay structure for the transmission of sound energy. 4. Tripus, the largest of the ossicles, directly connected to the anterior wall of the swim bladder, functions as the primary receiver of swim bladder vibrations. 5. Os suspensorium, sometimes included, a supporting structure connecting the ossicles to the skull and the otic capsule. Supporting structures. 1. Redi mirabilis gas gland, swim bladder. The swim bladder serves as a resonator, amplifying sound waves. 2. Ligaments. Ligaments hold the ossicles in place and allow precise movement transmission between them. Function of the ossicles. Ossicle claustrum, position closest to the inner ear, function transmits vibrations from other ossicles to the ear. Ossicle scaphium, position between claustrum and intercalarium, function links the claustrum to the next ossicle in the chain. Ossicle intercalarium, position between scaphium and tripus, function acts as a connecting ossicle for sound transmission. Ossicle tripus, position largest, connected to swim bladder, function receives and transmits vibrations from the swim bladder. Connection of Weberian ossicle with air bladder and internal ear. 1. Connection with the air bladder. The swim bladder acts as a resonating chamber for sound waves. When sound waves travel through water, they create pressure fluctuations that cause the swim bladder to vibrate. The Weberian ossicles transfer these vibrations to the inner ear. The tripus, the largest Weberian ossicle, is directly connected to the anterior wall of the swim bladder. The vibrations from the swim bladder are transmitted to the tripus, initiating the chain reaction through other ossicles, the intercalarium, scaphium, and claustrum. 2. Connection with the internal ear. The internal, inner, ear of fishes comprises 3 main structures: the utricle, saccule, and lagina, which contains sensory hair cells that detect vibrations and movement. The claustrum, the ossicle closest to the otic capsule, is linked to the ear structures. Vibrations transmitted through the ossicles reach the utricular otolith, a calcareous body in the inner ear, where sensory hair cells detect the mechanical movement. The inner ear converts the mechanical vibrations into nerve signals sent to the brain via the auditory nerve for sound processing. Ear-air bladder connection types. Type of connection: direct connection. Examples: herrings, shads. Key feature: swim bladder touches the inner ear. Type of connection: Weberian apparatus. Examples: carps, catfish. Key feature: sound transmitted via Weberian ossicles. Type of connection: lateral connection. Examples: eels. Key feature: lateral swim bladder extensions. No ossicle involvement. Type of connection: no connection. reduced/absent, examples sharks, rays, flatfish. Key feature: no functional link between swim bladder and inner ear. Different types of caudal fins. The caudal fin, tail fin, plays a critical role in propulsion, maneuverability, and stability in fishes. Based on the shape and structure of the lobes and the vertebral column's orientation, caudal fins are classified into 1. Homocircle caudal fin, structure, symmetrical lobes, upper and lower, with the vertebral column ending near the center of the fin. Example, most teleost fishes, e.g., salmon, trout. Function: efficient forward propulsion with minimal energy expenditure. 2. Heterocircle caudal fin, structure. The upper lobe is larger than the lower one, and the vertebral column extends into the upper lobe. Example, sharks, sturgeons. Function: provides lift as well as thrust, important for buoyancy. Control in cartilaginous fishes. 3. Difficircle caudal fin, structure, symmetrical and tapering, with the vertebral column extending to the tip. Example, lungfishes, coelacanth. Function: efficient in slow, continuous swimming. Modifications of caudal fin. Caudal fin shapes are specialized for different swimming strategies. 1. Forked caudal fin. Example, tuna, mackerel, adapted for continuous, high-speed swimming with reduced drag. 2. Lunate caudal fin. Example, swordfish, marlin, crescent-shaped, providing maximum speed in fast swimmers. 3. Rounded caudal fin. Example, clownfish, angelfish, suitable for maneuverability but inefficient for sustained speed. 4. Truncate caudal fin. Example, bass, grouper, balanced between speed and agility. 5. Pointed caudal fin. Example, some eels, helps in slow, undulating movements. Modifications of caudal fin. Origin of paired fins. The evolution of paired fins, pectoral and pelvic, is one of the most significant events in vertebrate history, leading to the development of limbs in tetrapods. Theories of origin. 1. Gegenbohr's archipterygial theory suggests that paired fins evolved from archipterygium, a single lobe-like structure similar to the limbs of tetrapods, supported by the structure of fins in lungfishes and coelacanth. 2. Lateral fin fold theory proposes that paired fins originated from continuous lateral folds along the body of ancestral fishes, supported by embryological evidence where lateral fin folds form before differentiating into pectoral and pelvic fins. 3. Gill arch theory, Balfour and Thatcher, suggests paired fins evolved from modified gill arches, largely discredited in favor of the lateral fin fold theory. Significance of paired fins. Pectoral fins aid in steering and maneuvering. Pelvic fins contribute to balance and braking. In tetrapods, paired fins evolved into forelimbs and hind limbs, facilitating terrestrial locomotion. Thank you for watching.